Hello, my name is Alex Ticho, and I'm an MD PhD student at the University of Illinois Chicago in the Department of Physiology and Biophysics. I'm the lead author of the manuscript titled A Novel Bioluminescence-based Method to Investigate Uptake of Bile Acids in Living Cells, which was recently published in the American Journal of Physiology. Thanks for watching this short summary of our manuscript. Bile acids are synthesized from cholesterol in the liver and secreted into the intestine, where they perform a variety of important physiological roles, such as facilitating fat absorption. They are then reabsorbed in the distal ileum and recycled to the liver through the portal circulation. Bile acid transporters, particularly the ileal apical sodium-dependent bile acid transporter, ASBT, and the hepatic sodium taurocholate cotransporting polypeptide, NTCP, are crucial for this recycling process. Recent evidence indicates that the activity of these transporters is rapidly regulated by phosphatases and kinases, as well as by their association with plasma membrane microdomains. However, current methods to measure transporter activity do not fully capture these rapid functional changes. In this study, we aim to design and validate a real-time method to measure bile acid transporter activity in living cells. This method relies on a novel probe consisting of a cholic acid moiety conjugated to firefly luciferin via a cleavable linker, which we synthesized in high yield. The conjugation site on the bile acid side chain was chosen to conform to the structural requirements for ASBT substrates. Once the probe is transported into a cell, cytosolic glutathione cleaves the linker, leading to the release of free luciferin. In cells that express luciferase, luciferin will be oxidized and light will be produced proportional to the amount of probe that has entered the cell. This light can then be detected by a sensitive IVUS camera. We transfected HEC-293 cells with luciferase and either ASBT or NTCP, while the serotonin transporter SERT was used as a negative control. Cells were then incubated with the probe and immediately imaged by IVUS. Imaging was performed continuously for 45 minutes with a 1-minute exposure time. Luminescence production was then quantified using living image software. Our results showed that bioluminescence production was dependent on the expression of bile acid transporter. Bioluminescence was significantly higher when increasing amounts of ASBT or NTCP plasmids were transfected in HEC-293 cells. Additionally, when ASBT-expressing cells were treated with protein tyrosine phosphatase inhibitor 3, which has been shown to enhance ASBT activity, bioluminescence was correspondingly increased. We also performed studies using intestinal epithelial cells isolated from transgenic mice that constitutively express firefly luciferase. IECs from the jejunum and ileum were incubated with the probe and imaged for 5 minutes. Ileal enterocytes displayed significantly higher luminescence than jejunal enterocytes, consistent with the expression of ASBT. In this study, we have designed and validated a new method for measuring bile acid uptake in real time and in living cells. This method has many potential applications for in vitro use, including to identify drug compounds that alter bile acid uptake. Future studies will focus on applying this method to in vivo systems, which should allow for non-invasive, real-time quantification of bile acid absorption in live animals. Thanks for watching this video summary, and I hope you take the time to read our manuscript.